Okay, so this is a little quick video to show you how to take coordinates from one genome build for some of your locations of interest, and probably you have them as a bed file, .bed file. There's different versions of bed files, but you might need to convert. In this instance, this is an example of here's some contents from a bed file. You can often open these up just in Excel to look at them easier. They're text files and they're probably tab delimited. But every bed file, no matter what version you have, there's 3 minimum column information requirements. In this case, the first column has to have the chromosome number. The second column will have the starting coordinates of the feature, and the third column has to exist to tell you the ending coordinates of the feature. And this is just base number on the chromosome. Optional is a fourth column and some other columns, but you often might have a fourth one there. And the name, for instance, of your gene might be put into there. This is for a capture panel. So these are all areas of the genome that are being captured for targeted sequencing. And this has actually been done in what is called HG38 build of the genome. And so I want to convert this over to HG19 coordinates because we are going to use an Illumina alignment system module called an enrichment module, and it happens to just be scripted to look for an HG19 genome on the machine and to use that for the alignments. So I need these coordinates in a different build of the genome. And so this is how to do that. If you go to the genome browser at University of California, I would just go to the home page for that, and there's different tools available. This one is Liftover. And what you can do, and in my case, you can see here I can pick an original assembly I want to convert coordinates from, so I have them in HG38. I'd like to go to human genome HG19, and so that's available for conversion. And you will put them into this window or upload the bed file, or you can paste them in. Just to show you kind of how that would work or not work, let's use a paste in some of these. What you'll find out right away is, even though this is information that was in something called a bed file, if you use this format in here and submit it, it will have 4 records that all basically displayed errors. And what's happening is it can actually convert it, but it turns out that what's happening is exactly how you had this chromosome designated. And so it turns out that, at least in the current version of the tool that you access at the genome browser site, what I've done is made a copy of this tab, renamed it HG38CHR. What I've had to do is put in CHR instead of just 1. So you need CHR to designate the chromosome. So if I go between the two, you can see the difference. And it's not too bad. Paste it in a few places and drag it down, and then just be careful to get your chromosome numbers all correct. So this information is okay. And this is the way to use the conversion. So I can save this tab as a text tab delimited text file and upload it all at once. But I've got 817 entries here. This is all pastable as well. So what works in this case is, once I have it in this format, I can grab all of these 817 entries . I'm going to copy, and this should just paste actually into the window. Pretty straightforward. It all looks like it's arranged properly all the way down. I think all of them are there with no problem. I can now submit this. This time when it processes, it's successfully converted all 817 records. good. If you want to see them, you do the view conversions. It actually downloads another bed file, which I'm going to open up, and TextEdit just opened it up for me. This is what you get back, and those I have pasted on to an HG19 tab here, and you can see going between the two builds, it's in the same place. If I just flash between the tabs, all that's changed is there are different coordinates in each build. But now I have the HG19 bed file. This returns another number, one designation in another row, which you don't necessarily, I don't necessarily need that. I've just took this file, contents, I just pasted it into another tab back in Excel, deleted the extra row of ones here I don't need. So now I have a bed file. I can generate a bed file from this, which is now my HG19 version of the coordinates, and we'll use that to make a manifest. And so we can either use this to align to HG19, and if we want to align to HG38, we've got that as well.